Hello, everyone. Uh, Welcome to today's webinar. I'm Mengqi Wang. Today's webinar I will introduce you something about single-cell omics. This is the contents of today's report. I will begin with a brief introduction to the background and development of single-cell sequencing. Then I will introduce you our BGI Genomics single-cell sequencing product, the DNBelab C-series single-cell RNA sequencing. The first part is the background. We should first clarify what is single-cell analysis. When we talk about sequencing, we usually refer to the bulk sequencing. The bulk sequencing obtains the average information of genes at the tissue level. In contrast, single-cell sequencing provides the gene expression information at the cellular level. When it comes to the spatial sequencing, it can help us obtain location information. So the bulk sequencing always analyze a large number of tissue cells together and then get the average result obtained may mask significant differences between individual cells. However, even in semi-uniform populations, cells can vary greatly in gene expression, protein levels, and behavior. Also, single-cell analysis overcomes this limitation by examining each cell individually, revealing subtle variations in genes within different cells, and thereby uncovering biological meaningful heterogeneity. So that's why we need single-cell analysis. When we use single-cell analysis, we understand cells in dimensional ways, like qualitatively, we gain the diversity of cell types generated through the regulation of gene expression. Quantitatively, we gain the count, proportions, and density of each cell types, and also the spatial levels. We can gain the spatial arrangement of each cell type and their interactions within cellular ecosystem. Also, the temporal level, we can gain the specific timing of changes in cell types and states. Then the directional level, we can gain the potential transformation passes each cell type may undergo. So understanding this multifaceted dimensions of cellular diversity and organizations is crucial for comprehending the connections between the genome and the specific biological processes, as well as the regulatory mechanisms involved. What can we do with the single-cell analysis? Single-cell analysis can help us with various fields by providing detailed insights into cellular functions and interactions. It's crucial for oncology development targeted cancer therapies. And in developmental biology, it can help to understand cell differentiations and embryonic development. Additionally, single-cell analysis can also help to, in the notation of spatial omics. Also, we can also integrate multi-omics data for comprehensive biological analysis. So single-cell analysis offers a powerful tool for advancing research and applications across multiple scientific domains. The study of cell biology can be traced back to the invention of a microscope. In 1990, researchers demonstrated the feasibility of single-cell transcriptomics. In 2009, single-cell high-throughput sequencing was introduced for the first time. From that point on, single-cell sequencing became a powerful tool, allowing researchers to identify new cell types and investigate the mechanisms of disease at the single-cell level. By 2018, a commercial platform has emerged, and in 2019, BGI Genomics launched the high-throughput single-cell sorting platform, the DNBelab C series. So this will be the focus of my upcoming report. The first will be the the first part will be the workflow of DNBelab C series single-cell RNA-Seq. First, this is the overview of the workflow of DNBelab C series RNA-Seq. At the beginning, we need to obtain a single cell or nuclear suspension. Then we use the droplet generator machine. We call it a TAM4 to perform cell sorting and barcoding. After droplet breakage, molecular barcoding amplification, we will harvest the cDNA. Then the cDNA will be used to construct the library and the sequencing. Finally, we use this data to do the bioinformatic analysis and visualization. So this is the strategy of our product, DNBelab C series single-cell RNA sequencing. The technical principle is based on the microfluidics to achieve high throughput. There are two critical indicators in single-cell RNA sequencing. The first is the captured cell count, and the next is the median gene count per cell. You can see on the website, on the report of website, also the estimated number of cell. So the capture cell count of our platform can be range from 8,000 to 10,000, and the median gene count per cell can be 1,000 to 3,000. The actual number is depends on the sample types and the quality of the sample. Next I will let me go through the workflow step by step. And at the very beginning is the sample prepare step. Single cell sequencing is a high cost project. So strict control during the quality control stage can help us obtain high quality data and save costs. The sample preparation procedure is the decisive step of the experiment. The left part is an overview of sample preparation. Our services can start both from the tissue dissociation or prepare the single cell suspension. When obtaining the cell suspension, we would check for the debris. We need the cell viability to be about 85%, and the cell number should be at least 100,000. Obtaining the high-quality cell suspension is very critical because dying cells are easily to lyse to release the RNA. The release of cell-free RNA will cause background noise in the detection and affect the quality of single-cell data. So it's a sample preparation. Then we will use the high-quality cell suspension to do sorting and barcoding. The sorting is based on microfluidics. The cell suspension and beads will flow through the channels and are wrapped into oil droplets. The cell beads combine with the cells and then capture the RNA. After reverse transcription, cDNAs will be released and used to construct the library. So here is. Sorry. Here is the structure of the library. The cell barcode, the green one, is used to label cells, and the polymerase T, the purple one, is used to capture mRNAs, and the UMI, the blue one, is used to label mRNA. The sequencing is performed on the DNB-Seq platform, and the sequencing strategy is paired and 100. Then we would capture about 8,000 to 10,000 cells and generating about 600 million raw data. When sequencing finished, we need to perform bioinformatic analysis. The accompanying software package, DNB C4 tools, providing fundamental analysis, including data quality and cell clustering. So it's a demo report of DNB C4 tools. We can also do the standard and advanced analysis on our platform, Dr. Tom. The standard analysis including data quality control, single cell gene quantification, cell clustering, cell type annotation, and cell function analysis. And the advanced analysis including differential analysis of multiple samples, cell communication, RNA velocity, pseudo-time, and CNV analysis. So this is the Dr. Tom. Dr. Tom is an advanced and intuitive data analysis platform. We can do the analysis I mentioned before on this website. It can Dr. Tom can enable the interaction of cell and gene data results, as well as dynamic and static analysis of interactive mining of single cell data, speeding up the publication process. You can analyze your data and create your desired figures faster by just clicking on the Dr. Tom website, without the need of complex programming languages like Python or R, without asking for the favors from your busy bioinformatician friends, without the need for software downloads. It's easy to load, faster to visualization, and you can perform all the analysis I mentioned before with just a few clicks on this website. If you want to simplify and customize your analysis, you need to try this. That's about is the workflow of the product. When we have a single cell platform, we need to check the function of the platform. So first, I will introduce you the sensitivity experiment of our platform. We perform the sorting using two types of cell lines with known ratio, as you can see here, PPSC and NPSC. The percent is 99 versus 1%. And as we can see, from the right pictures, trans types of cell sorting can be achieved, which is basically consistent with the present ratio of the 1% and the 99%. This reflects the high sensitivity of our single cell sorting platform. The next, we want to evaluate the technical reproducibility. Then the experiment use one PBMC samples, and the one sample was divided into 4 groups. You can see here into 4 groups, and the same bench was loaded on the 4 channels of our machine TM4. The cell profiles and cell types obtained show high consistency. So Our platform are also consistent with the published data. This page show you the one PBMC sample loaded on the TM4 and some published data on the on the paper Nature Biotechnologies in 2020. The notation are consistent. So this is the function measurement of our platform. Then I will show you some performance of the platform by entry data. This slide shows the data of a cell line sample and a PBMC sample. The input cell number is 8,000 and the capture rate both about 40%. The overall performance is also good. This slide contains several typical samples like human PBMC, human nucleus, mouse brain nucleus, and mouse cancer cells, and even some OCT samples. In this slide, you can see the overall performance of our platform is also good. So in this part, I show you the workflow and the excellent performance of our platform. Then I will show you the advantages of our product and even our BGI Genomics. BGI Genomics was the first to launch in the single-cell sequencing market at very early time and process rich project experience. We have our own single-cell sorting and sequencing platform. You can also choose many omics and different platform depending on your needed. Also, the Dr. Tom analysis system I mentioned before can help you accelerate the data analysis and the publication. Also, so we could offer you end-to-end services from the sample to the data mining and even the article. So that's the project advantages. And when we talk about the DNBelab series, it also helps many publications. From 2020, DNBelab series has helped published 140 articles with an average impact factor of about 12. So the following is some publications of this platform. Okay, in the next part, I will introduce the application of our product. As I mentioned before, we have DNBelab C platform has accelerated many publications. So I will show you many publication cases to show our product applications. We can use the single-cell sequencing data for many research because single-cell sequencing can help clarify the cell type composition and characteristics of different cell types. This is so-called cell antlers. We can also conduct a comparative analysis of different states and describe the dynamic changes process. Also, then we could find the causes of these changes. And this idea can be used in embryonic development, tissue differentiation, cell reprogramming, tumor genesis, immune stress, aging, and regeneration. As research depends, single-cell research has gradually entered the scale-up stage. In addition to antlers research, single-cell EQTL and single-cell large cohort research is increasing day by day. The development of multi-omics has also increased. Single-cell combined with the transcriptome and the genome, and also proteome and even spatial omics. So in the next part, I will show you some cases. This is the first application of C-series single-cell RNA sequencing. This article was published on Nature in 2022, explaining the technical details and the methods. So based on the single-cell library construction and sequencing platform independently developed by BGI Genomics, the research team conducted single-cell sequencing analysis on about 1.14 million cells. These cells are from 45 male tissues and organs of macaques. And the researchers divided these cells into 113 major cell types, and even divided them into about 400 cell subtypes. So this article constructs the first non-human primer single-cell antlers in the world, and this data can be get in the interactive website NHPCA. So this data can be used for the species evolution, human diseases, and drug evaluation and screen. This slide shows some cell clustering and annotation result of several typical tissues and organs. This is another case we published in Development Cell. This study combined single-cell RNA-Seq with stereo-Seq. Stereo-Seq is BGI's latest spatial sequencing technology. This study analyzed a total of 19 sagittal frozen sections in 6 key development time points within 24 hours of fertilization in zebrafish embryos. So this study also constructed a spatial temporal transcriptome antlers of zebra early embryonic development, and this data can also be gained at the website Zesta. So this is this study. The next study is an application in metabolic disorders. Researchers combine single-cell RNA-Seq and stereo-Seq, somewhere like the former one. They use the 16 to 18 years old macaques hypotheses, and the samples they set in 3 controls, 3 obesity and 3 diabetes. In total, they get nearly 500,000 cells. This study draws the spatial transcriptome antlers of hypotheses under obesity and diabetes. And then this study also revealed the important role of INF and PVN in maintaining the engine balance. This study is about tumor research. Researchers use the tissues from colorectal primary cancer tissue. We also call it the CRC. They use the CRC tissues, adjust the non-cancerous tissues and distinct normal tissues to do the single-cell RNA sequencing. In total, 15,000 cells were obtained, and a high-resolution immune landscape of CRC was drawn. So this study revealed the extensive changes in the proportions and function states of T cells and B cells in CRC tissues. We can also use the platform, also single-cell RNA sequencing to study the infectious diseases. This article is about high-resolution cell antlers of the pig's lung, and it also put the data on online platform for exploring lung single-cell data. In this study, it systematically studied the similarities and differences in cellular crosstalk and expression patterns of respiratory virus receptors in each cell type of pig lung. The next study, this study is about a large cohort of liver cancer. You can see from the sample number. The study used 160 ppmc samples from 124 cancer patients. Researchers defined 5 immune microenvironment subtypes of liver cancers with single cell precision for the first time. This one, you can see this one. And this classification system was named timer laser. Also, this study also revealed the heterogeneity of tumor-associated neutrophils. So this is the case of cohort. The next two study is about a single cell EQTL. In the first study, this one, authors analyzed more than many PBMC cells. These PBMC samples are from 162 SLE patients and 99 control, that's healthy people. Then the authors integrated dense genotype data to map cell type specific cis expression quantitative trilogy. We can also call it cis EQTL, and linked SLE associated variants to cell type specific expression. This study combined population genetics and single cell RNA sequencing to annotate genetic variants with cell type specific effects on gene expression associated with SLE. So the next study is performed on nearly 10,000 healthy people. The sample is also PBMC. Researchers identified many EQTL and cell type specific EQTL. This means they linked genetic characteristics to cell type specific gene expression. So using this data, the researchers find potential causes of some autoimmune diseases, and this data can also be get at the website. It is the address. So these two analyses study combine WGS with single cell sequencing, and they linked the genomic characteristic with the cell type and the expression informations. That's the analysis of EQTL. Okay, then the application of single cell RNA sequencing. Then the summary of my today's report. At the very beginning, we learn about the background of single cell sequencing. So the single cell sequencing experience the single cell high throughput sequencing. And then the commercialized platform can help sequence many cells at one time. And we BGIs also have our own single cell platform, the DNBelab C-series platform, and our product is the DNBelab C-series single cell RNA sequencing. So in the middle, we go through step by step the workflow of a single cell RNA sequencing. That includes cell suspension and the sorting and barcoding, and then we get the cDNA. The cDNA will be used to construct the library and the sequencing, and finally is the analysis of data. At the last, I introduce you the application of single cell sequencing as the research deepens the many cohort and EQTL analysis and studies emerges. So that's today's report. Thank you.